Welcome. Let's begin with one of the very interesting topics of science, and that is heredity and evolution. So these are two different concepts that we would be considering today. The concept of heredity being given by Mendel and the concept of evolution being propounded by Darwin. We would be understanding both the scholars, their work today in a very concise and a very simple and an interesting manner. So before we begin with, we have some of the brain teasers for you. Now if you are able to solve these, you are all set with the chapter. If not, let's proceed with the coming topic, and let's see if you are able to answer those as we proceed. Now, across the class, we would be talking about some of the important concepts. First is the contribution of Mendel. So, the P generation, F1 generation, F2 generation, monohybrid, dihybrid cross, monologous, analogous for your Darwin. And those are some of the key concepts that would be discussed in this class. Now, first important topic that we consider is accumulation of variations. Now, how does it happen? Any organism offbreeds or propagates. Now the propagation of an organism can vary. It could be by a single organism or it could require multiple organisms. So we broadly say this reproduction is either sexual reproduction or asexual reproduction. Now, when you have the offsprings being produced by a sexual reproduction, the variations are much higher, and that's where we talk about the accumulation of variation. Let's say you have one bacteria. Now this bacteria independently divides into two. These independently divide into further two, and this another into further two. But these four bacteria that are produced from a single bacteria would be very much similar to the original bacteria. As a result, I could say that there are very minor differences that are seen in the generations of bacteria due to very small inaccuracies that are present in the DNA copying. However, let's take an example of human being. So let's consider you, your parents, so your mother and your father, and then you have you yourself. You can understand the kind of variations in the physical appearance, in the genetic makeup, we could say. So the kind of the things that are required for a reproduction here or an offspring which is coming here is much more diverse. So the diversity varies with the kind of reproduction taking place. Also, let's talk about bacteria. So bacteria, if I can say, has a unique capability of standing extreme weather climatic conditions. So it could stand through the extreme heat conditions, extreme temperature conditions. That means you would have selection of certain traits or variants that are controlled by the environmental factor. Because if those traits or variants are not being able to pass through the changes of the environment, those won't be taken up with the next generation that is there. So, to understand this, we have the concept of heredity. Now, this concept of heredity was given by Mendel. Mendel's contribution is indeed interesting. He was a student of science and maths at the University of Vienna in Austria, but he could not complete it. Actually, he failed, and as a result, he went to monastery. But his interest into the science of hereditary could not stop him. He started experimenting with pea plant, and that was the sole thing that brought about the present day genetics. Now, why he first of all took pea? There were few reasons. It is very easy to grow. The lifespan is very short, so the results come very quickly. One species of pea plant is clearly distinguishable from another species of pea plant, and the flower size is bigger. Also, it's a unique plant where self-pollination can take place. So, you require a plant which can have self-pollination. So, we understand cross-pollination.
Pollination is a pollination that occurs between different plants. However, when we talk about self-pollination, it is a pollination that occurs within the same plant.
You have a new plant that could be seen. So, this is a unique plant where you can have self-pollination, and therefore, he chose pea plant for his study. Now when he chose pea plant for his study, he tried to understand that there are both paternal and maternal DNA traits that are present as in the human beings, and when we are talking about inheritance, sometimes let's take a very simple example to explain this. Let's say your father has a attached earlobe. So earlobe can be of two types. It could be directly attached to the skin, or it could be hanging, and those are called as free earlobes. Okay, so let's say your father has an attached earlobe, the mother has the free earlobe. Now what is the probability of the kind of earlobe that the child would have? So this is something, a question that can be answered through genetics, and this was what was explained by Mendel in his very basic concepts. So let's begin with the three terms, that is the P generation, F1 generation and F2 generation. P generation, as the name suggests, is the parent generation. F1 generation is a cross pollination or a cross of the parent generation. So you would have, let's say, a pea plant here, okay, and another pea plant here. What we are trying to do is trying to study the cross pollination. So for this cross pollination, what Mendel took was a pea plant with TT. So that's the tall trait, okay, and a small TT denotes the short trait. Now, while he was giving his study, what he did was he crossed these two. So when you cross these two, what would happen? You would have T, T, T and T. So all the generations would be what? All the generations, if I visualize, would be T and T. That would be the mix of these two generations, the parent generation. These are the P generations that are there, and this generation is the F1 generation. So all the four plants that we got, or all the four traits that we got here, have T and T trait. What does that mean? This means that all the plants by nature are tall, because the capital T is a dominant trait. So what he tried to explain was, every time you have a dominant trait and a recessive trait. Dominant trait is a trait which behaves or which dominates the scene, and as you can see, since you have even a single capital T here, this would be the dominant trait, and with one small t you would have a tall trait that would be present. So the new flowers that you would be seeing would be all tall in nature. Now this was the F1 generation. From P to F1 generation we come through cross pollination. From F1 to F2 we come through self pollination. Now how does it occur? Now I already have my F1 generation, which is TT. I self pollinate means I cross it with another TT. Now let's work on this. So this is the cross tabulation that we have. How does it work? You have capital T, capital T, capital T, small t, capital T, small t, and small t, small t. That means this is completely tall. These two are the mixed variants, but since T is tall, you have the tall species that is there, and this is the shortest species. Now how do you understand this? The very simple way is, we talk about two terms, phenotype and genotype. Phenotype means what is the physical appearance. So what is the physical appearance? I can say 1, 2 and 3 are tall, 1 is short. So phenotype ratio would be 3:1 for these four plants. However, the genotype ratio would be 1 tall, 1 short and 2 hybrid. So 1:2:1 would be your genotype ratio. This cross that we did leads to an F2 generation. So the resultants that we see here are of F2 generation. But what we considered here was just one trait, and that was tall versus short. And since we are considering only one trait, we call it as a monohybrid cross. Okay. Now, what Mendel did was, with this, he tried to explain a new concept where he used a dihybrid cross. So he believed that let me take two characters instead of one, and let's see how it proceeds. So in the parent generation, he took two characters, that was R, Y, and small r, small y. Now the capital R denotes round, small r denotes what? Wrinkled. So one characteristic was round and wrinkled. The other characteristic was capital Y denotes yellow, small y denotes green. Okay. Now what he did was he crossed these. When he crossed this, so this was the parent generation. The first cross would result into capital R, small r, capital Y, small y. And this was the F1 generation. This F1 generation was self-pollinated. So when I pollinate this with the same, what I would get? I would have R, Y, capital R, small r, capital Y. Okay, so that's how you have the 4 elements. But I would combine these elements. So let's say RY, capital RY, and small r, small y. So I would have capital R, small y, and small r, capital Y. Okay, and here what would be the traits would be? Again capital R, capital Y, capital R, small r, capital Y, and small r, small y. Okay, this is capital. Now when you cross this, how many conditions you would get? You would get 16 different conditions. Of this, only one condition that would be here, which would be small r, small y. And this is the wrinkled and the green condition. There would be 3 conditions where you would have small r, capital Y, small y. Again you would have small r, capital Y. You would have small r, capital Y, small y. And these 3 would be the condition where it would be wrinkled, because small r stands for wrinkled. Okay, so these would be wrinkled and green. So small r, small y would be wrinkled and green. So only one case for that. Then you would have this as wrinkled and yellow. So wrinkled and yellow, I have 3 cases. Now the next case would be, I would have capital R, small y, capital R, small y, and then I have capital R, small y. So these are the 3 cases where I would have R and small y. So that means it would be round but green, and all other cases would be the remaining cases where it would be round and yellow. So 3 cases, 1, 2 and 3, which are round and green. 3 cases which I mark as cross as wrinkled and yellow, and 1 case which is wrinkled and green. The rest all are yellow and round. So what is my phenotypic ratio? Phenotypic is that what I can see, it is 9:3:3:1, and that's how we understand a dihybrid cross. But if we complete this and you see the genotypic ratio, that genotypic ratio would be very different. As in the case it was 1:2:1. So here again it would be 1:2:1, then again 1:2:1. But in the between you had these 2:4:2. And that's the complete sequence of the genotype that you would study further in the higher classes. If you want to solve this Punnett square, so this square that we draw is what is known as Punnett square. And when you solve this Punnett square, you would be able to have this genotypic ratio. So we talked about the F1 generation, which is also known as the first phylalial generation or the cross pollination of the parent generation. F2 generation is the second phylalial generation or the self-pollination of the F1 generation. And then we also understood the law of segregation. So under law of segregation, what Mendel tried to state that there are 2 factors that control the formation of the gamete, okay, and these factors which bring in a specific characteristic help the plant to segregate. So it could be tall, it could be short. So understanding the dominant and the recessive trait is something that's very, very important here. Now, As we said, there are two traits. One is inherited, that is we get it from our parents. The other is acquired, which we get it through the environmental learning process. So those two traits are understood. The second trait, the acquired trait, is what we understand later. But what is the mechanism of inheritance? The mechanism of inheritance is very, very simple. It is controlled by the genes. So each gene controls one character, we could say. There are more than two forms of the genes that are present. Again, one form of the gene is dominant over the other form. The gene that is dominant is transferred to the next offspring in a higher probability ratio. So let's say in the case of tall and short, we had two genes that are present for the same trait. Now, of this, the dominant one takes the presence. Genes are present where? Genes are present on the chromosome, and within the genes you have the DNA. Every individual can have the similar genes or dissimilar genes. If they have similar genes, there is probability of similarities that could be seen at a genetic level, which could be revealed at a phenotypic level. That's the level of appearance that we could say. Okay, and when you have the two genes or the two forms of the gametes that are brought together, you have the fusion that takes place and you have zygote formation process that occurs. Now, how does the traits express themselves? As we said, you have the DNA structure, which we would study in detail at the further classes. So within the genes you have the DNA. DNA is the cellular information that is present and that is used for making the protein of the cell. Now, each section of the DNA provides one protein information which is called as gene of that protein. Okay, so the DNA that provides the information for one protein is called as the gene of that protein. Now let's talk about the height of a plant. The height of the plant could vary based on the hormones that are present in the plant. Okay, so based on the amount of the hormone, we can say the height of the plant would vary. So if the hormones are higher, the height might be higher. If the hormones are less, the height might be less. And thus, genes control the characteristics or what we call as traits. So individual characteristics or traits are controlled by what? These are controlled by genes. Very, very simple. Now, whenever you have a offspring, it's the parent genotype that determines the genotype of the offspring, and therefore we say both the parents have a role in determining the offspring. Now, when we say each germ cell must have one set of the gene that is present, we understand the determination of the sex. You have the male and the female. So in human body, you have 23 pairs of the chromosome, of which 22 pairs are similar. You have one pair that varies. And that pair in the male is XY and in the female it's XX. Now when you have the cross, how would this happen? So it's XY, XX. So you would have XX, XX, XY and XY. So this would determine whether the child would be a male or the female, and that trait comes in through the parent or the father, we say. Okay, so that trait, whether the child would be a girl or a boy, is determined by the father, and that's the inheritance that we study in human beings. Now coming on to a very interesting case. Understand this case very, very carefully. So when we talk about evolution, what we understand is three cases we would discuss today. The most interesting part of this class. So there was a group of beetles that were present. All the beetles were red in color. Case 1. There was some variation in the beetles and some green beetles started to come. Now when the crow came, the red beetle was very easy to catch. As a result, the crow could easily catch the red beetles, but during that time the crow was unable to catch the green beetles because green beetles were well camouflaged with the grass. So green in color and green in color. So for the crow, it became very difficult to catch the green beetles. As a result, over the time, what happened? The population of the red beetle declined because the crow was feeding on them, but the population of the green beetle started to grow, and this was the process of natural selection which was given by Darwin. So Darwin propounded the concept of natural selection, bringing this very simple idea into application. So initially we say that Darwin and Mendel did remarkable works, but both of them were contemporary, but their work was not shared among themselves. So if it would have been shared, the discoveries could have been much further, we might say. The second case that Darwin took was, there was a group of red beetles, there was some color variation due to some reasons, and there was a blue beetle that was introduced. So over the time, you had a huge population of red beetle with few blue beetles that were there. But suddenly you had a group of elephants that entered, and all the red beetles there were destroyed or killed. Now what happened? You had only few species of the blue beetle that remained. But over the years, these blue beetle grew, and the number of the blue beetles increased. So what kind of population came in was the blue beetle. But this case too was not a case of natural selection. This case was an accidental survival, because there was an accident where all the red beetle got destroyed. As a result, what happened was, only few beetles that were there were of blue color, and these blue beetles started to evolve, and you had the offsprings which were blue in color. So it led to a genetic drift. Drift means a shift. So it led to a genetic drift, a diversity without adaptation. So there was no adaptation from the red beetle that was there. Red beetle vanished off, and you had a new species of blue beetle that evolved. The third case is pretty more interesting. So these two cases are different. The third case is further different. The third case, what happened? There was huge population of beetle that was there. So they ate lot of grass. As a result, the amount of grass decreased. Now when the amount of grass decreased, these beetles start to starvate, and they grew very lean and thin. Now what happened over the time? There was good monsoon and a lot of grass back. So over the years, what happened was, these beetles were able to have the grass again, and the health condition improved. So the third case is totally based on food availability, and it is the adaptation of the species. So low weight is not a kind of trait that could be explained by inheritance. Okay? So that was something that he tried to speak about. Now we talk about two traits, inherited traits and acquired trait. Inherited trait is a trait that we get from our parent generation. Acquired trait is because of the condition. So because there was no grass, what happened was, the beetles starved and they grew lean and thin. But as soon as the grass was there, they grew healthy and they were back to their normal size. So that's how you have either the inherited traits or the acquired traits. Now let's say a very interesting example is, you have a huge population of mice with good length of tail. Now what happened? There needed to be some experiment done on mice, and as a result, surgery was performed on them and the tails was cut. Now what would be the offspring? The offspring would definitely have the long tails, because the cutting of the tail or removal of the tail was a surgical process, and it was not an inherited trait that was seen. So we are not bringing any change into the germ cells. Okay, unless and until we don't bring the change into the germ cells, it won't be transmitted to the next generation. That is very, very important. Now when Darwin was studying, he was only 22 years old when he went for a voyage, and to his voyage he took 5 years to reach South America, and after he got back to England, completing his voyage, he never left the shore and he worked continuously on the process of evolution and natural selection. So Darwin's theory of evolution is very interesting. He was also a naturalist because he conducted studies on the role of earthworms in the soil fertility. Also, we talk about certain characteristics which are passed from the parent generation to the offspring. We could say the color of the eye, the texture of the hair, okay, the color of the seed. So those are some of the traits which are transferred from either the plant or the animal species from the parent generation to the offspring. But there are changes that occur in the lifetime due to the environmental factors, and those are not transmitted to the next generation. For example, there could be a loss of finger due to accident, and that is something that is a kind of acquired trait and is not transmitted to the next generation. So what we need to understand is another important criteria where we talk about how things evolved. Now, how things evolved could be explained in a very simple process of the formation of Earth. So when we talk about formation of Earth, you had a huge land body that was there, and you had the water body. Slowly and gradually these continents drifted apart and you had the individual continents. But at that time we realized the species that were seen in the regions of Africa was very similar to what was seen in South America because of the close proximity at one point of time, and due to the drift that took place, you had the genetic drift that took place, and the same species was seen at two different regions. The next thing is how organic or organism arise. Now Darwin's theory tells us how life evolved from a very simple process to more complex process. Mendel gave the mechanism where he talked about inheritance of the trait from the parent generation to the offspring and further later. It was Haldane, a British scientist, who suggested that life developed only from inorganic molecules that are present, and the first primitive organism that originated was a result of a chemical synthesis process. Later on, you had experiments given by Miller and Urey that talked about that life exists only through the interaction of ammonia, methane, hydrogen sulfide, and there was no oxygen that was present over water. The temperature was below 100 degrees and this mixture was passed through lightning. When it was passed through lightning, the carbon was converted into simple compounds, and then later on you had amino acids that were formed, and from this amino acid you had protein molecules which were formed, and from this protein you had the origin of life that was registered. So there were different theories propounding the origin of life. Some believed that from the simple to the complex organisms it evolved. Some believed we evolved from an inorganic to an organic evolution that took place. Now, the next is speciation. Speciation means you have, let's say, many species of beetle that are present, coming back to the same example. Now, there could be various cases. Each of the beetle species cannot fly for long distances, that's for sure. Some of the beetles accidentally from this group went into another group, and as a result there was interbreeding that took place between the two groups. So there was some development that was registered, or some change, some variation that was registered. But in one very interesting case, what happened was there was a population of beetle here, a huge mountain, and another population of beetle. Now this population of beetle which was here was pretty different from that beetle population which was across the mountain. What happened one day was an eagle came, it picked up a beetle and it flew across the mountain. By mistake, that beetle dropped down and it was alive. Now, when this beetle came in this set of population, there was interbreeding that took place, and because of this interbreeding, a very different species came up, and that's how you have the gene flow. Now it's very simple. As the distance increases, as we saw, the genetic constituents changes. So distance increases, the proportion or the probability that the same genes would carry over there would be very, very less, and the variations would be very, very higher. And as a result, in olden times it was recommended that further the marriages across the same community beyond the same gotra, it is considered much better, considering the same principle of genetics. So one population which is here, another population which is here, if there is a cross breeding between the two populations, the resultant offspring would be much more variant, much more different, that would be seen, and you would have a genetic drift that would be registered. So speciation may take place when variations is combined along with geographical isolation, as we saw. So there was a mountain in between, there was geographical isolation, and you had one species here, another species here. Definite variations were there between the same between the beetle only, and therefore the speciation was much higher in this case. The next is how we talk about evolution. So talking about evolution in itself is a very unique concept. We talk about two types of organisms or two types of characteristics. One is homologous characteristics and another is analogous characteristic, as we talked about in the brain teasers. So homologous are those which have the same basic structure but perform different functions. For example, birds, reptiles, amphibians all have this same basic structure of the foot, that we would say, but their function for using the foot is different for all the three. So birds use it to catch the prey. You have reptiles for movement, you had amphibians with webbed feet, that would be seen. So each of those, the functions would vary, but the basic structure is same. That's the same structure of the leg that you would see for the reptiles, for the birds, for the amphibians. Predominantly there could be minor variations in it. The next is the analogous organisms. The analogous characteristics, not the organism, sorry, analogous characteristics. Now analogous characteristics means the basic structure is very different but the function is same. A very good example is a bird and a bat. Bird and a bat both use their wings to fly, for sure. So the function is same, both are flying. But the birds have their wings made up of feather. On the other hand, for the bat, it is the skin and the folds of the skin which helps it in flying. So the structure is very, very different, but the function is same. When the function is same, we say it is analogous characteristic. When the structure is same, we say it is homologous characteristic. Over the years, we have seen lots and lots of evolutionary process. We talked about dinosaurs, we talk about corals now. So there have been a definite change in the structure of life forms across the years. Now, how do we find out that there existed dinosaurs at any point of time? We can check that out only and only through the fossil remains. And how do we find that there are fossil remains? There are two ways of finding it. One is the relative way. Relative way means the species which were present very prior in age groups got deposited in the lowermost layers of the soil. The newer species on the layers above it, further newer species on the layers above it, and further newer species on the layers above it. So let's say you had dinosaurs 20,000 million years ago. So dinosaurs at the lowest. Then you had another species of horses after that which got extinct. So horses above it, and so on and so forth. That's one way of understanding the fossil remains. The next way of understanding the fossil remains is through the carbon dating process. So you have the dating process through which different isotopes are used of the same element in the fossils, and through which we detect the age. Recently you had Rayoli in Gujarat, where you have the dinosaur museum that has been established. The region was considered as one of the largest nesting sites for dinosaur. Now fossils represent few things. First is the preservation. through which we are able to understand an evolutionary trait. So how the organisms evolved over the years, we can very well understand through it, and how it was established through the various time periods. So evolution by state is very, very important. A good example is cabbage. So cabbage, we consider it as only cabbage, but it's not a cabbage that we eat. There are so many variants where you have very short leaves closely tightly packed, we say it's cabbage. When you have big flowers which are having a arrested growth, we say it's broccoli. When we say you have a swollen part that comes up, we say it is kohlrabi. When you have very large leaves which are present, we say it is kale. Now all these are basically the variant of the same cabbage. Minor variations that occur through the process of natural selection, either based on a specific geographical region or based on the evolutionary history, and therefore you have different variants of the cabbage that are found. Those are cauliflower, broccoli, kale, kohlrabi, cabbage. So those are all variants that we are able to understand. Also in this process, we understood that sometimes at a point of time it might be a case that feathers, when initially the birds had it, it was meant only for the insulation to protect the bird from the cold. But later on, these feathers might got evolved as a utility or to help the bird to fly. So there was a natural selection or a progression that was seen. Also, at the same level, we talk about yet another interesting term, and that is molecular phylogeny. That means the approach is based on the idea that organisms which are distantly related, okay, listen carefully, the organisms which are distantly related as the two species of beetle across the mountains would have or accumulate greater number of differences in the DNA, and that's where you have the speciation that would be flourished. Okay, the next important thing for evolution is not equated by progress. So it's not that how the species has progressed over the years would help you understand that the species has developed. We say that we evolved from monkeys, but that's not practically a correct statement, because human beings and chimpanzee evolved from a common ancestry, and that common ancestry was the point from where you had the chimpanzee that came in and the human beings that came in. So evolution is simply a generation of diversity that could be seen, and the shaping of that diversity is governed by the various environmental factors surrounding it. So inherited trait, acquired trait and natural selection, all of these together determine how the species move. So evolution cannot be said to progress simply from a kind of lower form to a higher form, or from a simpler form to a complex form. It's not as simple as that. Evolution means a variety of phenomena that have taken place, new species that have emerged, and how the things have changed over the years. So even if we talk about human evolution, it's not that simple. We talk about variations in the race. So we talk about variations in the skin color, variations in the size of the nose, the texture of the hair. So all those variations, the head index, so cephalic index is another, nasal index is another criteria. So all these traits help us to understand how organisms have evolved, and evolution of human beings itself is in itself a separate branch which is known as anthropology, where we talk about human evolution. So this was a very fundamental class where we talked about the basic principles of genetics. We talked about the laws propounded by Mendel, so the P generation, F1 generation, F2 generation, the monohybrid cross where we use one character, dihybrid where we use two characters. Then we came up on to Darwin, his theory of natural selection, how the things varied, both the scholars who worked forward towards the discoveries, but they were independent in their thoughts. There was no mutual exchange of information that took place. Also, we talked about the acquired traits, inherited traits, the homologous and the analogous characteristics during the process of evolution, how the role of fossil fuel is very, very important to understand the process of evolution. So with this, we wind up this class here, and we'll be bringing many more interesting classes on science. So stay tuned, do subscribe, have a wonderful day ahead.